There are lots of you signing up for the 9th, and there is one brave soul on the 16th. You can sign up for one of those 2 days in February. And remember, you do have to pick a topic when you sign up. So it looks like I've got a third of you signed up. Other announcements, we will have our first lab on Monday, barring some unforeseen weather weirdness, which I'm hoping has cleared up by then. So lab is at 2 on Monday. I don't know if it'll go all the way till 5, because our scheduled test slot, in 204 on campus. And there is lab, the lab handout is posted on Moodle, so keep that off if you want. Where we left off, we're in this lecture that was posted from Tuesday. I had a few slides I didn't get to. We're talking about signaling pathways, signal transduction pathways, and some typical versions of how this works for a cell to get a signal and then to move it inside the cell and then somehow passage the signal inside the cell, usually to the nucleus among some other places. We left off, before I stay on that picture for 4 minutes, we left off with hedgehog signaling. So we've been talking about how hedgehog signaling works. A hedgehog, this protein here that's the ligand, which is actually the signal, is one of these paracrine factors. What does that mean?
It's a signal from the inducer?
Yes, it's a signal from an inducer. And it means that it's probably been secreted by a cell and it's actually moving a little short distance before it's received, as opposed to 2 cells touching, which we'll talk about today. So a paracrine signal is secreted and it moves a short distance, as opposed to an endocrine signal. It's different because it goes through the bloodstream and can usually move quite a long distance. So it turns out for a hedgehog to be secreted properly and make its journey between cells, it actually has to be linked to a cholesterol molecule for proper secretion and for proper movement out to its signal target. So if cholesterol isn't around, or cholesterol has been blocked or inhibited somehow, hedgehog signaling doesn't really work. So this brings us to our example up here. This is a lamb that was born with cyclopia, which means that there is a centrally located eye, although there are some other things going on here too, with some of the facial features and the mouth and the ears. Hedgehog is particularly important in getting the face to pattern correctly into 2 sides. And as it turns out, there's a plant called the corn lily that grows in pastures, especially in Northern California. If pregnant ewes ingest this plant, it actually contains 2 chemicals, one called germine and one called cyclopamine, both of which interfere with cholesterol. So if the pregnant mother eats the plant, those compounds are circulating in her system, and there's a cholesterol problem, which means that hedgehog signaling isn't working properly during embryonic and fetal development of the calf that's going to be born. And this is one example. Some of the calves don't even make it to birth. This is one that actually made it to birth, although I think this lamb actually died shortly thereafter, because they really have some issues with development. But it's an example of how other factors can influence signaling pathways and how important they are. All right, the last slide I actually didn't have on your slides. I added this on after the fact, which is why you're like, where is it? The only point here is that, again, if you have mutations in these genes that are making proteins that are working together in the same pathway. So in the hedgehog pathway, there's hedgehog itself. But then there's patched the receptor, there's smoothened the buddy-buddy protein with the receptor. There are other ones downstream of that. If any of those are influenced, you usually get a similar phenotype. So the original phenotype from this, remember, was in fruit flies, the larvae are supposed to have these bands of little hairs on their abdomen called denticles. This is how the larvae is supposed to look. This is a hedgehog mutant larvae, so you can sort of see where the name came from. I think in this picture better. Like, oh, this one looks like a little hedgehog. This is another gene called dispatched, which is needed for hedgehog secretion from the inducer cells. But if you had a mutant in patched, or in smoothen, or in one of the other proteins that are in this pathway, you will actually see the same phenotype. I can't remember if I put more words on this or not. No. All right. That was all from last Tuesday. So now we're caught up. So just this recap, what we talked about was cadherins and cell-cell connections via cadherins. Then we talked about induction and competence. And then I gave you two examples of signaling pathways. FGF signaling, which in this case we talked about this receptor tyrosine kinase or RTK pathway. And then hedgehog signaling. At this point, actually, I moved into today's lecture, so we haven't talked about delta Notch yet. All right. Questions so far? You guys ready for more pathways? Yes! Before we do that, I actually have a couple of videos if I can put them play. All right, so this is our Moodle site. I found two videos talking about cadherins, since we talked about these early on in chapter 3, and they're actually, I think, really taking a few minutes to look at if I get them to work on this video. Just to refresh your mind. Uh-oh. Oh. Wait for it. Wait for it. All right. How do I make that bigger? Bigger? No. Oh. Well, apparently we have to view this and shape. No. I think we have better computer knowledge than me know how to make this bigger. Oh, I did that. Well, apparently we're going to watch it this size. But I'll turn down the lights and I'll turn up the volume.
After its translation from mRNA, the classic cadherin protein is transported to the cell membrane. The cadherin establishes itself as a transmembrane protein with distinct cytoplasmic and extracellular domains. As seen in this conceptual model, the extracellular segment, which is responsible for the cadherin's ability to link adjacent cells presenting cadherins of compatible type, includes 5 characteristic domains. These cadherin domains interact with domains on neighboring cells in a calcium-dependent manner. In the presence of calcium ions, individual cadherins pair up to form dimers on one side of the intercellular junction. These cis dimers then bind with other dimers on the opposite or trans side. When calcium is removed from the environment, the cadherin molecules fail to bind, and cell-to-cell adhesion is disrupted. The cytoplasmic tail remains within the cell and forms a complex with a trio of catenin molecules, E120, alpha, and beta-catenin. This molecular complex links the transmembrane cadherin protein to the actin cytoskeleton. The cadherin-catenin complex is also important in cell-to-cell adhesion, as cells lacking catenins or expressing cadherins without catenin-binding domains fail to adhere as normal.
Okay, I think that's the end of that video. So that one actually shows a little additional information from the slides in the lecture. You guys see me with the lights off, so that's a second video. Okay. We talked about them as binding like this, the two cadherin molecules come together like this and binding. They actually, it seems seems like evidence suggests they do bind and then sort of slide past one another to make a tighter bond. We didn't actually talk about that, and that's not shown in the images in the book or in the ones I put in lecture, but they do show that in this video. And then they talk about 3 different catenin molecules. We've just talked about them all inside the cell as beta-catenin. They talk about individual different ones. I'm just going to keep referring to beta-catenin. And that's actually more the way they have it labeled in the book as well. So that's a couple differences. But I think it's kind of nice to see things moving and joining the cells in a representation rather than just the dry pictures in the book. All right, video number 2 is talking about how cadherins work.
Junctions between cells take several forms. Adherens are the main adhesive factors in the form of junction known as the adherens junction. Adherens junctions originate when 2 cells expressing cadherin molecules of the same type come into contact with each other. Cadherin binding occurs at the point of contact, resulting in a punctate junction. Subsequent morphological changes bring more cadherins on both sides of the junction into contact, creating a stable junctional zone of uniform width around the cell's entire perimeter. In this way, large numbers of epithelial cells can organize into sheets so that cells only tightly associate with other cells presenting cadherins of the same type. During the neurulation stage of embryonic development, a population of ectodermal cells initially presenting E-cadherin molecules begins to fold inward. These invaginating cells begin switching their cadherin expression from E to N-cadherin, which is specific to neural cells. The process concludes when the N-cadherin expressing cells at the top of the tube meet and close, completing the segregation of the neural population from the ectoderm.
I think that one is so cool. All right, so these are actually on Moodle, if you want to watch them again. Or show them to your friends and family. Movie night, I know. Exactly. I'll do that just for a second. So this is the second half of chapter 3. So this is going to talk about a few more signaling pathways that are also both part of this paracrine signaling. So there's going to be a secreted molecule that's the signal, and it's going to be bound by a receptor, and there's going to be some intracellular events. So we talked about the Wnt family and the TGF-beta family. After that, I'm going to talk about delta notch, which is an example of juxtacrine signaling, which is a little different. And then finally, a little bit about how cells maintain their cell type and connections with their neighbors once they've decided what cell type they're going to become. In other words, they've differentiated. How do they maintain that? The signal comes in and tells them, become a certain type of cell. Once the signal is gone, how do they know to remain that type of cell? And at the very end, I have an example called the epithelial mesenchymal transition, or EMT. For whatever reason, developmental biology is full of acronyms. So I'm going to try to explain them for you guys. Be forewarned, there's a lot of abbreviations. Sad to say. This is actually showing actin filaments in a migrating cell, which is kind of nice since some of these signals that come in will manipulate the cytoskeleton into changes. This is all actin. So the most polymerized actin filaments are in blue. So they're at sort of the edge of the cell, they're actually kind of like along a surface. And then the intermediately polymerized ones are in green, and then there's a pool of unpolymerized actin that's in red. So it's all actin. This is in different colors, which I think is kind of this picture. Like, takes these images. It's awesome. All right. I used to have, there were 4 different colors of pen in here. And so I didn't bring my own, and now there is one lone red marker. So we're going to see how I do with just red. Uh-oh. You have another one? Sweet. Oh, at least have some diversity here. All right. So what we've talked about so far. Is hedgehog signaling. Oh, I just forgot the other type of signaling we talked about. Thank you. And FGF signaling or receptor tyrosine kinase pathway. Tuesday. Now we're going to talk about something called the wingless or Wnt. This is Wnt pathway. And TGF-betas. These are going to be the four main paracrine signaling pathways. All right. So wingless was the original gene that was discovered in Drosophila. Anyone have a guess as to what the mutant phenotype is? I know, right? Like, at least that part's straightforward. So they discovered these flies that would develop without any wings, which I have to say, a Drosophila without its wings looks extremely odd. They look like ants crawling around. It's very strange. So it turns out the disruption of this gene inhibits wing development, and it actually produces other things developmentally. And then they later discovered there are actually 6 other wingless type genes in Drosophila. And then they looked for homologs in vertebrates, and they actually found 19 total Wnt genes in vertebrates. So typically these are called wingless in Drosophila and Wnt in vertebrates. And this is part of what's known as the Wnt family of these are secreted paracrine factors like the others we've talked about. In case you are really gung-ho about Wnt genes, there's actually a database where you can check out all the different Wnt genes in different organisms and what they do and how they're related, which I will not ask you anything on exams and so forth on anything except what's in lecture. But, you know, extra information, it's always good to tuck away. So in vertebrates, it turns out one of the places that Wnt genes work is in development of the kidney. So this is a normal, I think they've taken this from mouse, a normal kidney setup. So the adrenal glands are up here, these are the kidneys, and then there are the developing gonads down here. If you have a developing system from an animal that's actually deficient for one of the 19 Wnts, in this case Wnt4, the adrenal glands develop, but this is where the kidney should have been, and you can see the gonads are developing. There's actually some issues with gonad development too, but this is just an example of one of the places that Wnt signaling happens. All right, the pathway itself. So fortunately, these all sort of start off similarly. There's a secreted signaling molecule, and it's bound by, a transmembrane receptor that's at the cell surface. And in this case, inside the internal part is linked to a protein called disheveled. All right. In the situation that there's no Wnt signal around, so if disheveled is, there's no Wnt here, disheveled is here, and it's actually going to target a protein called GSK3, which keeps APC, another protein, and beta-catenin together. So in this picture, we're over here. GSK3, APC, this pink thing is beta-catenin. There's another protein in here called axin that I'm actually not going to talk about. And when GSK3 is keeping APC and beta-catenin together, what happens is that beta-catenin gets degraded. Okay. So this is actually what's happening. I should take Wnt out for now. This is what's happening with no signal. I apologize if you guys already drew this in here. You're like, so beta-catenin is actually going to be the molecule that's going to carry the signal into the nucleus. So if beta-catenin is being degraded, what happens to the signal? Okay. So in other words, if there's no Wnt protein, there's no signal that goes into the nucleus. The interesting thing about this pathway is that a lot of these proteins have actually been talked about other places. Beta-catenin we've already talked about as an anchor in cell adhesion. This is actually a second role for beta-catenin, and you're going to find that there's a number of proteins that have more than one role inside the cell. Vertebrates especially tend to multitask their proteins for different uses, depending on who their binding partners are. APC, you may have heard about. It's actually also a tumor suppressor protein. And APC is one of the genes that seems to be routinely detected in colon cancers. When you have colon polyps forming that become cancerous, almost always, when colon cancer has developed, if you do a biopsy of those cells and you look at them, they have a mutation in APC. So it also has some role in cell proliferation and cell division. Although here, it's being actually used as part of this Wnt signaling pathway. So it's another multi-use protein. This is going to be somewhat common. All right, I'm going to use my other color here. So back to the signal. When Wnt signal is bound, what happens is that disheveled becomes repressed. Well, disheveled becomes activated and actually represses GSK3. I should draw that better here. Have that idea. Okay, so the green is with no signal. The red is when Wnt is around. All right, so what I've drawn is that when Wnt is bound, disheveled is actually activated, and it's going to repress GSK3, which means that beta-catenin can free itself from this complex and not be degraded. So far, so good. Did that change up here bother you guys? I'll go over this again. And when beta-catenin isn't being degraded, it can actually move into the nucleus and act as a transcription factor. And here it's partnering with another protein, but it can increase transcription by binding to gene enhancers. So overall, the green part is when there's no Wnt. In this case, the receptor frizzled hangs out with another protein called disheveled, but it's not activated, which means GSK3 can actually keep beta-catenin and APC together, which means beta-catenin is going to be degraded. And that's the end of that. That's all the green part when there's no Wnt around.
So disheveled isn't actually doing anything when it's not active?
Correct.
It's just leaving GSK3 to do
Correct.
Okay.
Correct. That's why I changed the arrows around here. Actually, I can just do this one.
Where did it come up with the name frizzled and disheveled? I mean, is there a naming
Yeah.
standard in biology
So,
somewhere?
There's a naming standard in each organism. So what comes up usually is if they get named in fruit flies first, they're named after the mutant phenotype. So wingless, the signal here was named after fruit flies. Later, they discovered that frizzled receptor, this wasn't in fruit flies, I'm trying to think what organism this was, but the mutant phenotype when this receptor protein wasn't working was that they had a very frizzled appearance. And I actually don't remember what organism it was. Same, if this protein wasn't working, they had a very disheveled appearance. And this is like cellularly disheveled. I mean, I think of like frizzy hair and disheveled hair, but this is like whole organism disruption. So frizzled and disheveled are actually very similar mutant phenotypes. So if this isn't working or this isn't working, you get this sort of very perturbed embryonic development. Yeah. Then what happened is they realized these proteins work together in a pathway, and so when they put them all together, these names go with them. If they're named first in vertebrates, they're more likely to have a three-letter name that's an abbreviation for something else. So WINT comes from vertebrates. I can't remember what WNT. It's like wingless, integrated, and something else, other genes that were all in this family. So there is no naming standard. So the names are weird. I agree. Yeah. All right. When WINT signal comes around and it binds to frizzled, disheveled is activated, which is the marks here. And it can repress GSK3. In that case, beta-catenin can free itself from this complex and move into the nucleus and work as a transcription factor. So overall, when you have a signal coming in, you get an effect on increasing transcription of target genes.
What would you say the end result was?
It increases transcription of the target genes. So here, on this picture, it just says transcription. There are several downstream target genes that have an enhancer for beta-catenin, and they'll all be turned up transcriptionally.
You need to know about the
What's that?
LES and TPS.
Yeah, I'm going to ignore those. They are actually a partner transcription factor that binds with beta-catenin. All right, this actually is known as what's called the canonical WINT pathway. In other words, the typical or basic WINT pathway. It turns out there are some modifications that can happen to this pathway for other uses, which the book talks about, but I'm not going to cover because I think this is complex enough. But if you're reading about this in the text, this is the canonical WINT pathway. For whatever reason, biologists really like the word canonical. They use that a lot. Okay. Questions? You guys good? Yeah.
Just to clarify, we want the disheveled to be inactive.
Well, it depends on what you want. If you want to increase transcription of these genes, you actually want disheveled to be active. So when the signal is present, aka WINT, disheveled is active. The odd thing about it is that when it's active, it's repressing GSK3, which itself is a repressor of beta-catenin. So when you have sort of a repressor of a repressor, it's like you've activated beta-catenin. I don't know if that helped or confused further. But it's like a double negative here, which ends up being a positive in terms of the signal.
When it's blocked, are you getting when it's flying? Or is it when it's bound that you're getting?
Yeah, no, when this is blocked, like when there's no WINT, that's when you get wingless flies. All right. One more pathway. These are, I would say, a little painful to learn now. We will come back to these pathways and talk about them when we actually get to spots in development that use these signals, pathways to send a signal. So they will come up again in later chapters too. All right. This slide, don't be too overwhelmed by it, because I only want to point out a few things. TGF-beta family. And this includes a couple of different sort of subfamilies, actually just families, but this is referred to as a superfamily, but subcategories of proteins. One is a group of proteins that are all different versions of something called TGF-beta. So TGF is actually transforming growth factor. So this is a family of different proteins. They all have pretty similar structure that all work as growth factors in cells. Which usually means when you add them to cells in culture, it triggers the cells to either grow larger or divide. There's also a group of proteins in this superfamily called BMP proteins, which is bone morphogenetic protein. I put the words up here. And then there's a Drosophila category called decapentaplegic, which is abbreviated DPP. The only point I want to make about this slide is to introduce these names of the gene families and just to show that they are all related sequence-wise, amino acid sequence-wise, and structurally. These proteins all do similar things. There are 4 different TGF-beta proteins. There's a couple of ones called activins, which we're actually not talking about. And then there are all kinds of what we call BMP proteins, or decapentaplegic, which is actually the Drosophila version of BMP proteins. I know the names are weird. There's others in this family that I'm not going to focus on, but they're kind of funny. So there's something called VG1. There's something called screw, which is a Drosophila protein. There's something else called nodal. And then there are some others down here. So these are all proteins from different organisms on this tree that have very similar sequence and probably do very similar types of functions in the cell. They all work through the same type of signaling pathway, which is what I'm going to show you guys next. All right. All right, are you guys okay if I erase the Wnt pathway? I used to have this professor in college and he would say, the eraser is poised, fearsome felt. It's like, dear God, I mean, it struck fear into my heart, like I should write badly whatever he had up there, because it was about to leave. All right. Okay, so the TGF-beta pathway. What's happening is up here, they're showing sort of generalized function, and over here they're showing a couple of examples. This is going to involve the signal molecule, which is actually TGF-beta, or one of those TGF-beta family proteins. And what happens is when the signal is around, there are 2 receptors that can dimerize. They come together and sort of joint bind the signal. So again, this is happening at the plasma membrane of the cell. The receptors are actually slightly different. There's a type 1 TGF-beta receptor and a type 2. And unfortunately, they don't have a better name than that. So they're just type 1 and type 2. What happens is when the signal is present and they come together to jointly bind the signal, they cross-phosphorylate each other, which is what's going on here with all these phosphate groups. What other pathway does this sound like so far? Yeah, it sounds a lot like the RTK pathway. This one is different in that the phosphates are on usually either serines amino acid or threonines. These are serine threonine kinases that cross-phosphorylate each other. What amino acid does the RTK pathway phosphorylate? Tyrosine, that's the T actually in RTK. But other than that, they're actually very similar so far. What happens when they're phosphorylated? Is that they trigger phosphorylation of a protein called SMAD. And that name, I'm trying to think, it's a combination of other names that they've sort of like mushed together into one name, and I can't actually remember them at the moment. But there are actually different types of SMADs.
So they don't phosphorylate SMAD, but SMAD, they trigger the SMAD should be phosphorylated?
They phosphorylate SMAD.
They do it
Yep.
themselves. Okay.
And then once SMAD is phosphorylated, it can actually bind to a second SMAD, which just sounds weird. But what's happening here is depending on exactly what the signal is. So in this part, the signal here is an actual TGF beta molecule. In that case, the target that's phosphorylated is either SMAD2 or SMAD3, and those both, once they're phosphorylated, bind with SMAD4. If the ligand or the signal is something different, in this case it's BMP, one of these bone morphogenetic proteins, then the target is either SMAD1 or SMAD5 that binds with SMAD4. So in other words, there's a whole collection of proteins that are called SMADs, and they're all working in this pathway. Exactly what the signal is within this family of signal molecules determines which SMAD is phosphorylated that then binds with SMAD4. All right, once you have one of these SMAD family proteins phosphorylated and has bound to SMAD4, the whole thing goes in the nucleus. And it can actually either enhance or repress transcription depending on what the target genes are. All the other activation pathways we've talked about have increased transcription of target genes. In this case, they sometimes activate a target gene and they sometimes repress a target gene. And the difference is it depends on what enhancer they're binding, and it depends on what SMAD is in complex with SMAD4. But that's it for this pathway. So this one is actually, I think, one of the most straightforward pathways. Yeah?
So when it says SMAD 2 and 3, SMAD 2 and 3 didn't bind together, it's SMAD 2 or 3
Or
that
3.
binds to 4.
Correct. Okay. And I'm actually not going to ask you guys the numbers of the SMADs. All I want you to know is that these are SMAD family proteins. One is phosphorylated that complexes with another, and then they can move into the nucleus to become transcription factors. All right. That's the last paragraph pathway. Yeah?
Is it significant that among the two type
Receptors?
Type 1 and
Uh-huh.
type 2, that one of them has one loss?
Yeah, it is, although I have actually simplified it. So I'm not going to ask you guys specifics about how many phosphorylation points there are. As it turns out, there is good evidence that receptor type 2 actually gets fewer phosphorylation, phosphate groups in phosphorylation than does receptor type 1. That's actually true biologically, but for our purposes, I'm going to simplify that somewhat. Yeah. Good eyes. I like that. You guys are detail oriented. That's good. I know. You're like, I wish we were. Okay. All right. Let's take a little break there, because then the next couple of slides, we're going to change direction a little bit. So this is the end of all these pathways. So these are the 4 that I'm going to cover and hold you responsible for. In as much detail as we talked about them in lecture, there is extra detail in the textbook that might be helpful for you to read But if you read that and there's some other details we didn't talk about, don't stress about them. Let's take a little 5-minute break. You can stretch and run around. And snack.
Is that kind of the way this is all of this? I think stuff like reading the chapters and reading stuff.
Yes.
Okay, one of these
Absolutely.
beeped. That one's no screen.
They're going to be large text size.